podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience from Harvard University, where I focused on neuroendocrinology and the neuroscience of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to parse and translate the information guests share on the show, and in addition to the podcast, I write long-form content and send out a free weekly newsletter at the Mind and Matter Substack at mindandmatter.substack.com. I also offer private, science-based health consulting, where we work one-on-one and apply the same rigorous, evidence-first approach you hear on the podcast, but personalized. We can obtain and analyze your biomarkers, work on diet and behavior strategies, review supplements and drugs that you might be taking, or anything else related to general metabolic health. So, if you'd like more tailored guidance on your personal health, head to nickjikomes.com and fill out a short form or book time with me there. A great way to support my content in all of its forms is simply to visit mindandmatter.substack.com. You'll find a link to that in the episode description, and you'll also find a link to my current affiliate partners through my support page. These are products that I use myself that relate to physical, metabolic, or mental health. For example, one product I use every day is the AquaTrue water filter, which removes heavy metals, microbes, endocrine disruptors, you name it, so that you have higher quality drinking water. All of those links for health consulting, for my affiliate partners, and the Substack page are all in the episode description. So thank you very much for listening, and don't forget to check out mindandmatter.substack.com for all of my content. Thank you very much for joining me.
Oh, thank you. Nice to see you.
Do you want to just tell everyone a little bit about who you are and what your background is?
Yes, absolutely. I might give a little bit of a longer version so that it kind of introduces the field and what we're going to be talking about today. So I did my PhD in Europe between Switzerland and France, and actually that's where I started to study lipid signaling network. In Switzerland, my PhD was more into the physiology of lipid signaling, and therefore it was about energy dependence and from cell types to cell types, neurons and glial cells. And then in France, it was much more pharmacology, and it was much more around how different stimuli will actually produce these lipid signaling. And it turns out that these lipid signaling I was studying is arachidonic acid, which is one of the constituents that form the endocannabinoids. So it was a very opportunistic, good foundation for me. And then what happens is that as I was getting ready to go to do my postdoc, the endocannabinoid field was starting. It was the very beginning. They had just discovered the first endocannabinoid, Anandamide, which also has this arachidonic acid moiety. And I had the opportunity to go do my postdoc in San Diego with Daniele Piumelli. And I remember his question. He said, when we started, he said, if there's one endocannabinoids, there's probably a second one, because we know that a lot of, neurotransmitter systems and neuromodulators have multiple ligands and multiple receptors. So that's how I started into the endocannabinoid field during my postdoc. And it was very centered on 2-arachidonylglycerol, again, the arachidonic acid moiety. And then that was the very beginning. So we had so many fundamental questions to ask. How is it produced and everything where? And then I had the opportunity to start my lab, and I was really interested in trying to develop novel therapeutics. And, the field back then, the cannabinoid field was very much focusing on studying the side effects. Memory impairment would be a really big one. But I was really interested in trying to figure out if there had any therapeutic potential for the endocannabinoids. So I started my lab in Seattle at the University of Washington in the School of Medicine in 99, if I remember. 25 years ago. And then through all these years, what I've done is kind of navigate the field, trying to understand and identify the most promising therapeutic approach to target the cannabinoids. So Initially, I was interested in neuroinflammation, so multiple sclerosis. Then I got really interested in seizure maintenance, especially with the effect of cannabidiol and the FDA approval. That was very promising. So I studied quite a bit of seizures and how endocannabinoids can actually modulate those seizures. And then more recently, when you want to develop a therapeutic approach, what you have to do is you have to think about the benefit, but you have to think about the side effects. What is the potential side effects of a therapeutic approach? So I've been really interested in trying to separate the side effects versus therapeutic effects of both THC and cannabidiol. And to do that, I might tell a little bit about the new technology that we're using to monitor these things. So that's what we're doing in my laboratory. As you can see, it's very broad. It's really the goal is to find the most promising therapeutic approach and understand it at the molecular level so that we can go all the way to human clinical trials.
And what are the endocannabinoids? Like what you've already sort of hinted at the answer here. They're fatty molecules, and we'll maybe talk about where they come from and how they relate to diet and things like that. But just as a class of signaling molecules, what is the characteristic of endocannabinoids? What do they generally do?
Well, endocannabinoids are really interesting and very special. They are actually quite different from most of the signaling mechanism that we have in the brain. You know, the field started with excitatory glutamate, GABA inhibitors, and then the field got really interested, and when I say field is in neuroscience in general, going beyond these neurotransmitters and looking at neuromodulators. And that's when the field started discovering and working on serotonin, noradrenaline, dopamine. And then the opioids showed up. And then the opioids, you know, those are small peptides that modulates. And now we have yet another signaling mechanism, which is actually driven and carried by those lipid signaling molecules like the endocannabinoids. So they're very special. They're very different than the other signaling mechanisms. And we can already say, we can start by emphasizing that there's two endocannabinoids. So how, like I mentioned before, there's anandamide, the first one, which means in Sanskrit means inner bliss, very appropriate name. And then there's the second one that I was involved in discovering, which is 2-AG. And even I think the field now has really recognized that these two signaling endocannabinoids, anandamide on one side and 2-AG on the other side, are actually doing very different things in the brain and have very different functions, even though they're both endocannabinoids and they act on cannabinoids. We're going to be focusing on the cannabinoid CB1 receptors in the brain.
Got it. So there's a bunch of different stuff in the brain and body, different types of signaling molecules, different classes, different structures, different functions. Obviously, they all have to be made out of something. Some of them are made out of things like amino acids in the building blocks of proteins. You mentioned, you know, the opioids are often peptides. They are basically little proteins. A lot of classic neurotransmitters or neuromodulators are basically amino acid-like molecules, and so on and so forth. But the endocannabinoids are basically fat-derived molecules. They're pieces of fats that ultimately come from fats that we consume. So they're fatty substances. Where are they stored and where do they come from in our brain? Like where does when an endogenous cannabinoid is used by a neuron to signal something, to do something, where is the neuron getting it from, literally?
Okay, so let's unpack this a little bit because there's quite a bit of concepts here. It is true that endocannabinoids are lipids, and therefore where they reside is in the lipid bilayer of the membranes, for example. And those are what we call hydrophobic, so fat-preferring molecules. So they have a hard time going into solution into water. So they're mainly into the membrane. It is true that diet will influence the composition of the lipids, but it's not a direct relationship between I eat something fat and then I produce endocannabinoids. It's just the overall over time.
Yeah.
I think we need to think about endocannabinoids that these small lipid signaling molecules that are released from the membrane in an activity-dependent manner. The cell, there's actually two, we can emphasize already another difference in the or characteristic of the endocannabinoid signaling system is that some endocannabinoids are going to be produced in a tonic manner. They're going to be there all the time, whereas endocannabinoids can also be produced in an activity-dependent manner, on and off, just like neurotransmitter. I stimulate something and then I stop stimulating. So endocannabinoids are produced on and off over seconds, very small time periods, but they can also accumulate for a very long time.
Yeah.
So coming back, go ahead.
No, I think that's important, right? So when we say that these things are fat derived, and when we talk about certain fat derived substances in the body, it's not that, if I go eat a meal right now and I'm eating some fat and I'm putting it in my mouth and it goes into my stomach, it's not that those fat molecules literally get turned into endocannabinoids right now, right after I absorb it into my body. It's rather that those fatty acids get incorporated into membranes, into adipose tissue, maybe burned for energy. They get distributed throughout the membranes of my body. And then later on, they may or may not become things like endocannabinoids, but that will only happen if a relevant stimulus comes afterwards. So what I eat is going to influence the fatty acid composition of my cells and my tissues. And what happens to me, the stimuli that are impinging on my body and what's happening internally, that will determine if and when those things later on get turned into endogenous cannabinoids.
Correct. Absolutely. That's a very good description. So coming back to the idea of how are they produced, this is actually a very fascinating molecular machinery that's quite specific. So there's several aspects to that. So they are in the cell membrane. So they are lipids, and then we have enzymes that are going to cut those lipids, hydrolyze those lipids that are complex, and release smaller lipids that are going to be the endocannabinoids. Those enzymes are going to be different if we're talking about the first endocannabinoid, anandamide, versus the second cannabinoid, 2-AG. Yet another actually concept to emphasize that these two endocannabinoids are going to be produced under different conditions by enzyme number 1, which is NAPLD for anandamide, and enzyme number 2, which is DAG lipase for 2-AG. So two different enzymatic pathway are going to release those lipids from the membranes. So that's already important to emphasize. Because lipids do not like to interact with water, there's actually very recent studies that have discovered some mechanism by which you can actually shuttle these lipids from one cell to the other. Because from one cell, it needs to go through the cytosol, which is hydrophilic water. So it needs something to help it. And I know you had Steve Laviolette that actually interviewed, and he talked about the fatty acid binding proteins.
Yep.
So those are actually really fundamental mechanism by which these proteins that have hydrophobic greasy pockets, the endocannabinoids are going to go into this greasy pocket of the protein. And like that, there's going to be a shuttle that's going to bring this endocannabinoid from one cell to the other. Now let's go deeper. Let's double click on the cell itself. You can actually have endocannabinoids that are released from the plasma membrane, and then they will go intracellularly because there's also some intracellular receptors, and they're going to be doing a lot of things. You interviewed Giovanni Marzicano, so he discovered the cannabinoid CB1 receptors on mitochondria. So now we're talking about an intracellular signaling pathway where endocannabinoids are produced from the plasma membrane, needs to go intracellularly, and the shuttle is also going to be a fatty acid binding protein. So that's one mechanism that is actually very special. There's no other signaling mechanism that uses these shuttles. And this is actually really important because we'll probably talk about this, but it will become a very important and interesting therapeutic target, those fatty acid proteins. The second mechanism that has been recently discovered, because the question is, Since these lipids do not like to interact with water, how are they going from one cell to the other? I mean, glutamate, GABA, dopamine, it's easy. They're hydrophilic. They just go through water. But in the endocannabinoids, so there's actually new research that's shown that in an activity-dependent manner, again, the cell actually releases these extracellular microvesicles, tiny vesicles of lipid that are fascinating because they actually travel from one cell to the other, and they contain inside quite a bit of information. They might even have mRNA that will actually change the phenotype from the next cell. But they also have, because they're microvesicle, they have a composition of their lipids that is very specific, and it turns out that they actually accumulate endocannabinoids. So now you have a packet of endocannabinoids that goes from one cell to the other, fuses with the next membrane. You have more endocannabinoids in the next cell, and therefore the cannabinoid receptors are going to be activated. So that's how endocannabinoids are thought to travel from one cell to the other via those fatty acid binding protein or microvesicles, or intracellularly also via those fatty acid binding proteins. So those are very different and unique mechanisms.
Yeah. So these endocannabinoids, they are lipid molecules. They are little pieces of fat. And basically, there are shuttles. So you can quite literally think of this almost like a city planner, like there's buses or
100%.
like shuttles. Like a protein has a water-soluble end and a fat-soluble end. And a greasy molecule, a lipid, like an endocannabinoid, can get onto the shuttle and be shuttled from one cell to a completely different cell, or within a cell from one place to another, such as from the outer membrane to the mitochondria. And so there's these little shuttles moving all over in different cells, moving these greasy molecules around to and from all of the different membranes on the outer side of the cell, and even within the cell in places like the mitochondrial membrane.
Absolutely. And the trafficking map is actually a really good analogy, because you have, I don't know, I think we already have like 10 different fatty acid binding proteins. They're going to have different abilities to transport either anandamide versus 2-AG. So there's another difference. There might be different shuttling mechanisms for anandamide versus 2-AG that go at different speed. And all of this tight regulation of anandamide versus 2-AG signaling is going to determine how much of the endocannabinoid is going to activate the cannabinoid receptor and where.
And what are these receptors? Let's go through them one at a time. So the main one, cannabinoid receptor 1, what kind of receptor is it? How common is it? Where is it? Is it mostly in the cell membrane? Is it mostly in mitochondria? Is this in very specific parts of the brain, or is this something that's all over the place?
Yes, so the cannabinoid CB1 receptors is what we call the G protein coupled receptor. So it's part of the family of those receptors that are typically modulated, for example, dopamine, serotonin. So it's a modulator receptor called G protein coupled receptor. So it's going to modulate cellular activity. Pretty much, let's go at each level. So at the molecular level, what it does is where is it expressed is actually very important. So cannabinoid CB1 receptors are predominantly expressed presynaptically on neurons. And what they do there is they regulate neurotransmitter release. And they are typically inhibitors of neurotransmitter release.
So in other words, what you're saying is at the point of contact between two neurons, the first neuron is sending a signal to the second neuron. The second neuron is receiving that signal from the first neuron. The first neuron, the one sending the general neurotransmitter signal, that's the one where the CB1 receptor will generally be.
So let's start again. So we have a neuron number 1, which we're going to be calling the postsynaptic neuron.
Okay, so yeah, you're saying postsynaptic is number 1, the receiver of the message. Yeah.
Correct, the receiver of the message. And then what it's going to do is the endocannabinoid is going to go back to the presynaptic and say, slow down, that's enough, shut off, which is why the endocannabinoids are considered retrograde, so from postsynaptic to presynaptic, inhibitors of transmission because they slow down transmission. That's enough glutamate. Endocannabinoids come on CB1 receptors and reduce glutamate release.
Yeah, so if a neuron's getting too much input or it's gotten enough input already, it's sending a signal back in the form of an endocannabinoid to the neuron, giving it that input, and it's saying,
correct,
that's enough,
that's
no,
enough.
no more needed.
Feedback, that's enough, negative, absolutely. And actually, so now let's go. So that's at the subcellular level, and that's the most predominant is CB1 receptor. There's a couple of more locations where CB1 receptors are expressed. We already mentioned on mitochondria. And so that's the intracellular CB1 receptor. And there its function is going to be to regulate mitochondrial function. And mitochondria does many things, including producing energy and substrates. So CB1 receptor has a role in regulating how much energy the cell will utilize. And then there's also, although much less studied, there's also some CB1 receptors on the soma, like on the main cell. And there they have a slightly different function according to the initial results, is that there they will actually signal all the way to the nucleus and change the phenotype of the neuron. And that's actually another very predominant role of the endocannabinoid signaling system, is to regulate the phenotype of the neuron. What kind of neurons are we going to have, and where are they actually connecting? Because CB1 receptors, I believe, is already expressed at the stage 2 cell after, really at the very beginning. And actually, the CB1 receptors regulate cell proliferation and cell migration. So the endocannabinoid signaling system, these lipids, when they activate and interact with the CB1 receptor during development, they'll actually regulate the phenotype of the neuron.
Yeah. So these things are very, very involved in not just what mature neurons are doing, but actually in the development and the setting up of cells and circuits itself.
Correct. Which is why this is a good moment to say, which is why it's a major concern about adolescent consuming THC, because now the THC is going to flood the brain that is still developing, and all the CB1 receptors are going to be interacting with THC instead of having a very precise directed endocannabinoid that tells the CB1 receptor go this way. So this is one of the hypothesis of cannabis use disorder, is when adolescents are consuming THC, THC is activating all these neurons, and therefore the wiring of those neurons is not precise enough anymore, because instead of following the endocannabinoids, the CB1 receptors are flooded with THC. And to the most vulnerable adolescent, this could actually, it results in cannabis use disorder, but some of these patients actually have schizophrenic episodes, psychotic episodes, because of these neurons that have been miswired.
And so when an endogenous cannabinoid, such as anandamide, gets used for signaling purposes, so it's this little fatty substance that ultimately comes from fatty acids in the cell membrane, how exactly is the cell producing it? Is it literally clipping off a piece of the cell membrane?
Correct. Yeah. Or yes, it's clipping off a subunit, a small piece of. Imagine the plasma membrane as a bunch of Legos, and then it's taking off one of the Legos and it's cutting it off and giving it to the next cell. Absolutely. The one thing that's really interesting to think about in the dichotomy between anandamide and 2-AG, now the field is realizing how different these small molecules. There's just a new paper that just came out by a very predominant lab showing that different behaviors. So now we're actually able to, this might be a good time to actually introduce the new tools we have to actually study endocannabinoids. So there's actually new technology that came up from some labs that are allowed to sense or detect endocannabinoids production in mouse brain in real time. And basically the technology is based on the cannabinoid CB1 receptor. And then in the intracellular part of the CB1 receptor, instead of having that G protein that does the signal transduction, they attach the fluorescent green protein, GFP. And now each time the endocannabinoid hits that sensor, the green protein flashes a green fluorescence. So now we're actually able to take these sensors, which are proteins, virally infect some mouse brains so that these sensors are expressed in specific neurons or specific brain areas. And now when a little mouse goes and does its little behavior, we can actually see in real time endocannabinoids being produced. I have no idea the emotion I had the first time I saw that sensor go up and down in real time from a molecule that I discovered during my postdoc 25 years ago, in a tube. And now I see that molecule being produced in real time in a mouse. So now let's come back to, now that we know that we have this new tool to sense endocannabinoids, and there's tools that can sense either the first endocannabinoids, anandamide, or the second endocannabinoids, 2-AG. And now we know that actually different behaviors will produce one endocannabinoid versus the other. A very novel concept. For example, aversive stimuli, something that will create fear, like, shocking, an electrical shock for a little mouse, that will actually create endocannabinoid 2-AG as detected by the sensor. By contrast, if you give a psychoactive drug, for example, nicotine or cocaine or ethanol, these psychoactive drugs are also going to produce endogenous cannabinoids, but with different dynamics. Some of them will produce more anandamide, some of them will produce more 2-AG. And therefore, now the field is starting to realize that these different behaviors and these different stimuli differentially activate endocannabinoids, anandamide versus 2-AG, and therefore, most likely different cellular responses, different CB1 activation on different cell types depending on the brain area. So that's where the field is at now. We're trying to understand how endocannabinoids are related to different behaviors and different stimuli so that we can actually foster that and maybe develop novel therapeutics.
So people often say that endocannabinoids are use dependent or they're made on demand. So there's mechanisms that allow the cell to say, okay, we need some endogenous cannabinoid right now. We can pull some from the membrane and use it right now for whatever the stimulus that's calling for it is. And so what actually determines, what are the main signals that start to determine when and where and how much of these things are made? Are these things highly dynamic? Are they highly different? Can different stimuli drive very different levels of production? And sort of give us a sense for the dynamics here.
Absolutely. It's a great question. And we're at the cutting edge of science right now. These are actually the kind of questions that the fields are asking. So these endocannabinoids are produced by different enzymes. So it'll really depend on where that enzyme is located in the cell, in the subcellular compartment. So if this enzyme is only in cell type 1, for example, the enzyme that produces anandamide, NAPE-PLD, will be expressed only in neurons A. And by contrast, neuron B will only express the DAG lipase, which produces the second endocannabinoids, 2-AG. You already there have a dichotomy of one cell versus the other.
Got it. So depending on the concentration and pattern of proteins, such as these enzymes in any given neuron, it can determine the cocktail of cannabinoids that might be produced. One neuron might produce only anandamide, or it might produce only 2-AG, or it might produce a combination.
That's correct. Absolutely. And that's the first level. So the enzymatic activity that produces the lipid. So now how are we going to regulate the enzymatic activity? It turns out that most of these enzymes are actually calcium dependent. Calcium is a fundamental signal transduction mechanism in cells that when a neuron, let's focus on neuron, when a neuron is activated, when it's depolarized, you have this influx of calcium that gets into the neuron, and that rise in intracellular calcium then will go and activate multiple enzymes, including enzymes that will produce the endocannabinoids. So that's the second level of regulation, let's say, of endocannabinoid. The first one is where is the enzyme? The second one is how much calcium is reaching that enzyme, and actually for how long. So that's the third level of regulation.
Yeah.
So now let's stimulate the neurons with two different types of receptors. One receptor that will be on and off very quickly, or even three receptors. One that's on and off very quickly, one that goes on for minutes, and one that goes on for hours, like a glutamate metabotropic receptor for minutes, an NMDA receptors for almost, for plasticity. So then you'll have the third level of regulation, which is, does this neuron express this receptor, nicotinic NMDA? And for how long is that receptor going to be activated, which will result in the increase in calcium, which will activate the enzyme that will release the endocannabinoid.
And for the non-neurobiology people that listen to this, one of the things to connect here is when neurons are active, depending on how much they're active, the pattern they're active with, depending on the ion channels they have, calcium is tied to that activity. So when the neurons are active, some ion channels allow calcium to come in. And so there's the important piece here is that some of these enzymes that produce endocannabinoids are calcium dependent. And so what that means is the level of activity, the pattern of activity that a given neuron is experiencing is going to determine then the pattern of endocannabinoids it makes, because calcium is sort of the dot that connects the enzymes that make the endocannabinoid to the activity levels that neuron is experiencing overall.
Absolutely. Now let's go to the other end of the signaling network. So we've produced the endocannabinoids now in an activity-dependent manner. These endocannabinoids are going to activate specific receptors. And now we need to talk about how we're going to stop that signal with enzymes that actually inactivate those endocannabinoids. So there's another here, very nice diversity of enzymes that are going to stop endocannabinoid signaling. For anandamide, we have this enzyme, which is called FAAH, which is a fascinating enzyme. So each time anandamide is produced and activates the CB1 receptor, then it unbinds the CB1 receptor and it's inactivated or hydrolyzed by FAAH. So that's the first endocannabinoid. And then we have another mechanism for the second endocannabinoid, 2-AG. 2-AG is produced, activates the CB1 receptor, and then it's actually hydrolyzed or inactivated by two different enzymes that are expressed in two different parts of the cells, most likely because they have different functions. So the most famous one is monoacylglycerol lipase, MAG lipase. This enzyme that hydrolyzes, inactivates 2-AG is expressed close to the CB1 receptor in the presynaptic terminal. That means that MAG lipase is really regulating the amount of endocannabinoid 2-AG at the level of the target. There's a second enzyme close to the CB1 receptor. There's a second enzyme, which is actually postsynaptic, ABHD6. And that enzyme hydrolyzes 2-AG where 2-AG is produced postsynaptically. So you can see how these enzymatic pathway from one cell to the other are going to regulate the signaling network very precisely.
Yeah. So the production of endocannabinoids from the fatty cell membrane is coupled to the activity of a neuron. How active it is going to be related to how much calcium comes in. That determines how much these enzymes start clipping the membrane to make endocannabinoids. And then those get deployed. And then there's another set of enzymes that basically turn off the signal and get rid of those endocannabinoids. So what all of this basically means is on both ends, you've got enzymes, you've got protein-based regulation that allows you to have pretty precise spatial and temporal regulation of this endocannabinoid signal. The cell can turn on and turn off the signal in very particular locations on demand as it's needed.
Correct. It's very similar to other neurotransmitter and neuromodulator systems. Glutamate is produced in an activity-dependent manner, and then it's removed from the synaptic cleft by a transport mechanism. Noradrenaline is produced, and then there's an enzyme that, or even an uptake system that removes it. So the signaling system in the brain is always an on-off. You need to produce, activate the receptor, and stop. And the endocannabinoids, that's how they do it. There's another aspect that's really fascinating in lipid signaling, is that actually these lipids, when I say that the enzyme is going to inactivate these endocannabinoids, that is correct. That means that let's take the example of 2-AG. So let's take 2-AG. So 2-AG is arachidonic acid, the fatty acid, and then there's glycerol. The enzyme, what it's going to do is going to cut this lipid and then release arachidonic acid. Therefore, it's not an endocannabinoid anymore. But now it's arachidonic acid. Arachidonic acid is very bioactive. It will actually produce prostaglandins and be involved in inflammation. Now let's go before endocannabinoids. Diacylglycerol is the precursor of 2-AG. Diacylglycerol is actually bioactive also. It regulates protein kinases. So the way to think about these lipid signaling is fascinating because there's multiple. As a lipid is transformed into another lipid, it becomes something else.
Right.
Before an endocannabinoid, it regulates kinase. Then an enzyme changes it, it becomes an endocannabinoid, it activates cannabinoid receptor. Then another enzyme changes it, it becomes arachidonic acid, and now it's involved in inflammation.
Yeah. So you can have a piece of a cell membrane and it becomes another type of lipid, and then another type of lipid, and then another type, but they're not merely intermediates. At each step, that molecule is now a new molecule that does something. And so it becomes very important now to think about, well, how many steps are there, but also how long do you spend at each step?
Correct.
Yeah.
And therapeutically, when you block one enzyme, you're not going to only block one thing. What happens? You're blocking the next step too.
Right.
So you have to think about those things. Absolutely.
Yeah. And this will probably tie into why, as I'm sure we'll get to, why designing drugs and designing therapies isn't as easy as we might conceptually like to think, because it's not just this one thing does one thing. Lots of things are pleiotropic. They do multiple things. They can turn into another thing very quickly. And there's regulation at all of these steps. And it's complex, literally complex.
Absolutely. And it moves all the time.
So how do we start to think about the way that the fatty acid composition, the overall fatty acid composition of our membranes in our neurons or anywhere else, relates to the sort of overall pattern of signaling molecules that get produced, including cannabinoids? And how do we think about how that relates to things like our pattern of dietary fatty acid intake? So like if I start eating a different level of polyunsaturated fat in my diet tomorrow, and I continue doing that for weeks and weeks, is that meaningfully going to shift the membrane composition of the fatty acids in my neurons ultimately? And is that going to in turn affect sort of the overall levels or the dynamics of things like endocannabinoid signaling?
I think there's some initial evidence that might be the case, but I would say that's probably not the main molecular mechanism that regulates the endocannabinoid signaling system.
And so let's talk about, I guess let's just focus on anandamide and 2-AG separately and together. But let's just take them one at a time. How similar are they exactly? And then what are the differences in terms of their actual structure, and then the general functional differences that they tend to have?
So the chemical structure is similar at one level, like this fatty lipid, the fatty portion of these endocannabinoids is the same. They both have arachidonic acid. And there's a little piece of additional molecule to arachidonic acid. In anandamide, it's ethanolamine, so it's a small. And then in 2-AG, it's arachidonic glycerol. That little piece of molecule at the tip of arachidonic acid is what is going to clearly differentiate anandamide from 2-AG. because they have the same arachidonic acid moiety. That little piece of molecule will make a big difference in the way the endocannabinoids are interacting, binding with stimulating the cannabinoid CB1 receptor. Here's another very big difference between anandamide and 2-AG is that anandamide, as it interacts with the cannabinoid receptor, it's only able to partially activate the CB1 receptor, which is actually very similar to what THC does.
Mm-hmm.
THC is a partial agonist. It doesn't completely stimulate CB1 receptor, which is probably why it stayed around, because if you fully stimulate CB1 receptor, you become cataleptic, way too strong. By contrast, endocannabinoid 2-AG, the second one there, is actually a full agonist at the endocannabinoid, CB1 receptor. So that's another difference. Partial activation, anandamide, full activation, 2-AG.
Yeah. And this would actually correspond to, so activation of the CB1 receptor actually has an inhibitory effect on a neuron. So partial versus full activation would then correspond to somewhat decreasing activity versus more strongly decreasing activity.
Correct. So now let's actually look a little bit further in the difference also when we compare with THC. Because when somebody consumes or inhales THC, THC is going to flood the brain and therefore activate CB1 receptor everywhere that it finds it. Whereas endocannabinoids are going to be produced in an activity-dependent manner physiologically where it's supposed to be. That's their role. The best example I have is they're going to be produced when I go for a jog because it's hurting, right? So it's going to be actually controlling my pain. And I'm a Neanderthal and being, a T-Rex is behind me. I need to continue even though it hurts so that I can survive. So endocannabinoids are actually also very parallel to the endorphins, the opioid system, which also are endogenous painkillers. So that's how one can actually think about the big difference between THC activating all the neurons and inducing psychotropic effects, the high, versus endocannabinoids being produced exactly where they're supposed to be produced. And therefore, we don't get high because these endocannabinoids are just doing what they're supposed to do, which is, for example, regulate pain.
Yeah. I think it's important. People often ask this type of question, or they have of me in the past, where it's like, why aren't we high all the time if we're filled with endogenous cannabinoids? But basically what you're saying is, you might have one synapse in one part of your brain that needs more anandamide and it cooks it up right then and there. That's not going to affect how much gets made at some other synapse in another neuron. It's the sort of point-to-point, synapse by synapse regulation of these things. Whereas when you consume an exogenous cannabinoid like THC, you're now putting it into your bloodstream and it's kind of flooding everywhere and hitting the receptors all over the place all at once.
Absolutely. Here's an interesting example that I think about sometimes is in the hippocampus. So this part of the brain that's involved in memory formation consolidation. There's quite a bit of CB1 receptors that are there. So what is the role of the cannabinoid CB1 receptor in the hippocampus? Well, let's think about what happens when somebody takes so much THC all the time during adolescence. One of the first impairment is memory impairment for people that consume THC. They forget things. Their energy level, it might not be as high. That's because the THC is actually activating all these CB1 receptors in the hippocampus and is messing up the memory formation. Now, what is the role of the endocannabinoids in the hippocampus? Well, during a day, my hobby is photography, so I kind of hear about that sometimes. During a day, we see about 2,000 pictures. But we don't want to remember everything. I want to remember you. I want to remember how you look. So I'm going to consolidate that. My noradrenaline is going to take that memory and consolidate that. But there's many things that happened today that I saw that I don't want to remember. I don't need it. And the idea is that the endocannabinoids are going to go and eliminate this memory. I don't need it. I don't need to remember everything. So endocannabinoids could actually remove some selective memory, which is why the side effect of THC is memory impairment.
Yeah, and that's, it's always important to remember that memory, the flip side of remembering is forgetting. You need both things in order for the memory and the memory consolidation to actually be functional. You simply can't and don't want to remember every detail of everything. That's actually going to be an impairment.
Correct. Which brings us to post-traumatic stress disorder. All the individuals that have such strong fears that they actually have a hard time to forget. And one of the idea is that by using they self-medicate with THC to maybe forget those memories that are so hard to remember.
Yeah, yeah. And people often talk about cannabis and cannabinoids in the context of PTSD. Maybe someone like a veteran has these traumatic memories. The idea is, right, you have the memory, you have the episodic memory, like you remember what happened, but the emotional power is sticking with you. You actually want to kind of forget or dampen that component, and the failure to, forget that, so to speak, is what the PTSD is in some sense. And so the idea would be, well, how do you actually dampen the emotional salience of the memory? And perhaps cannabinoids are one way to do that.
Could be one way to do that. It's the memory was so salient that was so much noradrenaline. It was so stressful that it was consolidated and remembered, and there was not enough endocannabinoids to forget it. And therefore you have post-traumatic stress disorder. The issue with treating with THC is that it has a very tight, narrow therapeutic window. Mm-hmm. It might have some medical properties, but it will have some side effects. And therefore, really, it's a medical decision with the doctor usually to be able to find a good regimen.
Well, so how do we think about things? We've already said that this endocannabinoid system, the system of endocannabinoids, receptors, and so forth, it's ubiquitous. It's all over the brain. It's not like in one part, and it's spatially and temporally specific, right? It's sort of a point-to-point regulation to this. You can use it in a synapse-specific manner. Given that, if someone with PTSD, let's say they have a traumatic memory that is bothering them and causing them issues in their lives, you want to dampen the emotional salience of that memory that they're failing to forget, so to speak. But you don't want to interfere with memory consolidation generally. You don't want to disrupt other memories in their brain. How do you start to think about playing with this endocannabinoid system so that you're going to selectively help with the forgetting and the remembering in the one circuit that's problematic, or I should say the one memory engram that's problematic, without scrambling memory or disrupting memory more generally?
Absolutely. That's where actually the small molecules or medicine that will modulate the endocannabinoid signaling enzymes might come in. The big difference, again, between the THC that is going to activate all the receptors throughout the brain. And actually, let's have one little precision. Not every neuron in the brain expresses CB1 receptor. There are some neuronal population that don't. And one of the most salient one to think about is the dopaminergic neurons from the VTA for that. And therefore even though there is actually a drug addiction component to cannabinoids, and then cannabinoids increase dopamine, right? It's pleasureful. There's an addiction. Actually, those dopamine neurons do not express CB1 receptors. The other ones close to the dopamine neurons that regulate dopamine neurons that stimulate dopamine production. But coming back to what you were saying, I think that's where the field has a lot of promises, is to actually target the enzyme that regulate the endocannabinoid signaling system instead of stimulating all the receptors. Because, for example, for memory is actually a process. You have your short-term memory, and then you have your long-term memory. So it would be an interesting testing to do would be to actually give some inhibitors of the enzyme that inactivate the endocannabinoids. So if I take an inhibitor of that enzyme, I'm going to have more endocannabinoids that are going to accumulate, and therefore maybe it'll help me forget. And I'll do it in a very specific time period where I have the short-term memory, and therefore that short-term memory is gone, and it's not sent to the long-term memory and consolidated.
I see. And I
that
guess
would be one way.
Yeah. And I guess if I play with this a little bit, the way that I'm imagining that would go would be, if we remember the fact that memories are consolidated and reconsolidated, every time we recall things, the memory sort of becomes a little bit more labile and can be changed, can be weakened, strengthened, changed in different ways. I guess the idea here might be that if you have someone with PTSD, say, they've got a traumatic memory, they're failing to dampen that memory, to forget the emotional intensity of it, you might have them, say, take a therapeutic, like an endocannabinoid-based therapeutic, specifically when they're recalling that memory, but not all of the time. And that might help it sort of be dampened down or forgotten, at least to some extent.
Absolutely. That would be nice. It'd be nice to try.
Now, how much of this is theoretical? Like these ideas kind of all fit together in a conceptually nice way, but have we seen much success with developing things like endocannabinoid enzyme inhibitors to actually show these kinds of benefits? Is that something people are working on today?
That's a good question. So there's a lot of hypotheses out there, very little theory. Theory will become, that's when we have something that's written in the textbook and we know it works like that. We have a lot of hypotheses and we are going to be testing that it's working like this. So now this might be a good place to start talking about preclinical testing of novel endocannabinoid therapeutics. So the cannabinoid field is actually quite young as compared to the opioid field, because we only discovered in the 90s the endocannabinoids and the receptor compared to the opioids, for example. So we know much less on the endocannabinoid signaling system compared to the opioid signaling system. So there is, and then there's been some challenges to do preclinical research in the cannabinoid, not much funding, and also the Schedule 1 limitation to have access to THC. All of this has really slowed down the field to make progress. Now things are a little bit better, so there's actually much more that we understand. So the preclinical field now, and we have the tools also to actually do some nice, thorough preclinical testing. So the field is actually now testing multiple things, multiple therapeutic indication. If we either, the most logical one to start with is the one that we talked about, is to target the enzyme that degrade endocannabinoids.
Yeah. So just so people are clear, there's enzymes that naturally degrade endocannabinoids when they're released. So they're controlling sort of when the signal gets turned off. One of the things that is attractive about making that a therapeutic target is you can regulate how much of the endocannabinoid signal there is, but still maintain sort of the natural patterns of endocannabinoid production.
Only where they're produced physiologically,
right,
you make it stronger. Absolutely. Absolutely. So there's some things that, so we're going to be talking mainly about two enzymes. We're going to be talking about FAAH, the fatty acid amino hydrolase that inactivates anandamide, endocannabinoid number 1. And then we're going to be talking about MAG lipase, monoacid glycerol lipase, that inactivates the second endocannabinoid 2-AG. So the first one, targeting the fatty acid amino hydrolase, unfortunately, the field went, in my opinion, a little bit too fast into humans. We didn't really have enough information. The two limits, there's two critical things that you have to think about when you develop a new therapeutic approach. One, you have to make sure that your molecule, your new medicine, is going to do the job that you expect. It's going to go and inhibit fatty acid amino hydrolase FAAH only, and not do something else that could be toxic. The second challenge is to actually find the actual disease that we're going to try to temper or even cure. And that's where the preclinical testing in our little mice, we have mice models of different diseases. And therefore, what we're going to be doing, the field is to take that FAAH inhibitor and test it in our little mouse for certain indication. What is it for pain, for anxiety, fear anxiety, for depression, all these indications. Unfortunately, there was actually a first molecule that was a FAAH inhibitor that went into a human clinical trial in Europe. And unfortunately, that clinical trial, or fortunately, was stopped right away because of tremendous side effects. Tremendous side effects.
What were they?
I believe there were some patients that were even considered brain dead. They had no more neuronal activity in their brain.
Wow.
A handful. Obviously, when you have that kind of side effect, you stop the clinical trial right away.
Right. And I guess so the idea here, right, if it's a FAAH inhibitor, you're inhibiting the enzyme that degrades anandamide. So you would expect to have more anandamide generally, which means more inhibition of neural activity. And basically Correct. You're saying if you take that to the extreme, you basically get
Well, actually, so there's the second part to this story, which is what I said first, is you need to make sure that your molecule is clean and it's doing what it's supposed to do. And what was discovered a couple years later in a science paper by my colleague Mario van der Stelt, he actually, how would you say, he profiled that molecule. He looked at what are the other proteins that are inhibited by this inhibitor that we thought were specific to FAAH. And he found this other protein, I forget what the name was, but it was a critical protein involved in neuronal viability. And therefore, this first molecule, unfortunately, that was given into humans, stopped the clinical trial right away. The entire field freaked out, obviously. This is a bad therapeutic approach. But then now we understand it's probably an okay therapeutic approach if you have the right molecule that only inhibits FAAH.
Yeah. So for some reason, people thought this was a specific FAAH inhibitor, an inhibitor of just this enzyme, but it wasn't. How does that, is that an easy thing to miss? Or was this just like a weird case? Why was it thought that this was specific?
I wish I was on the field to understand the details. But in drug development, that is a really big component that you do up front, is you kind of look at what are the off targets, what are the other proteins. Typically, what we do is we look at the off targets, the proteins that are critical for life. Some proteins in the heart. Don't give this molecule because you're gonna have a heart attack. That we know right away. I think this was a less understood enzyme that was targeted by this molecule, and therefore it was not on the radar. People were not thinking about it. And it's only when Mario did this unbiased approach that he discovered, oh no, there's this enzyme. So now obviously we know molecules should not target that enzyme.
Yeah, and I guess
they're going to be brain dead.
And I guess that's one of the strengths of being able to do unbiased approaches in preclinical animal models is you don't have, there's known unknowns, there's unknown unknowns. These unbiased approaches allow you to sort of see everything, even if you don't already know something about it.
Correct. So that's the story of the inhibition of FAAH. I think the field is still working on it and trying to have cleaner, more selective FAAH inhibitors, and maybe find the right indication. What are we going to try to cure anxiety? And now that we know in our little mice that different endocannabinoids do different things, that might help the field to find the right indication. And now there's other really promising therapeutic approaches that now are moving forward based on preclinical evidence and cleaner molecules, and they're targeting the monoacylglycerol lipase, the second enzyme that regulates 2-AG. And those are moving forward nicely. There's multiple indications, including anxiety and addiction. And I believe they're already in phase 2 human clinical trials, which means that their toxicity level is pretty good. There's not much side effects if they're moving forward. So maglipase, I think that's a very promising area. And then there's also, we mentioned the fatty acid binding proteins, those little shuttles that there's some inhibitors now that are in clinical trials also based on strong preclinical mouse studies. They found the right indications and they're treating these patients with these inhibitors of fatty acid binding proteins and boosting the endocannabinoids. The endocannabinoids often are, I like to call them the help me signal or the repair signal. They're produced when you need to slow down things and therefore that's the way I think about it in the therapeutic approach is what you're doing is you're boosting the endocannabinoids and therefore you're boosting the help me signal and you're helping your body to deal with the disease.
Mm-hmm, mm-hmm. What about, so oftentimes the nervous system gets the most attention when we talk about endogenous cannabinoids. But of course, these things are used elsewhere. They're used in the immune system. They're used outside the nervous system in a bunch of tissue systems. What are some of the bigger examples that help us understand what this system of regulation is doing outside of the nervous system? Why do we have these receptors and these endocannabinoids in various different cell and tissue types? And what does that sort of tell us about the core function? I think you were kind of just starting to allude to it a little bit, but can you maybe give some examples of, say, immune system regulation by endocannabinoids or somewhere else to help illustrate this?
Absolutely. So it is true that we have that the cannabinoid receptors are expressed by multiple cells in the peripheral tissue. I would say that if we start with the cannabinoid CB1 receptor, the level of expression of those CB1 receptors is much lower in those other cells in the periphery compared to neurons. So when we think about THC, we know that when you take THC, there's going to be some psychotropic effects. And THC will also modulate some physiological responses in the periphery. Immune cells is one, vasculature, blood pressure is another one, which is why individuals have red eyes when they're high, because there's a hypotension and therefore dilation of the blood vessels. There's quite a bit of cannabinoid receptors in the liver, and they're really involved in liver metabolism, which is why there's this really interesting inverse correlation between the individuals that take THC and actually lose weight and they eat more. They take THC, they have the munchies, and actually they do not gain weight. There's this dichotomy. It's because those CB1 receptor in the liver, for example, are going to regulate liver metabolism and energy expenditure and metabolism. So the liver, here's one organ that doesn't have cannabinoid receptor is the kidney. There's the muscle also doesn't really have, the heart doesn't really have many CB1 receptor. Good thing, right? We're not going to die from a heart attack. The immune system is fascinating because, so one of the function of the CB1 receptor that we mentioned, so in neurons, it regulates neurotransmitter release. Okay. And their phenotype. What do they look like? In immune cells, it's a little bit the same also. You have those CB1 receptors that are going to regulate the immune cell transmission, their ability to release immune molecule. Those fatty acid we talked about, those prostaglandins, some cytokines. And here, the CB2 receptors are also very dominant. So immune cells have both CB1 and CB2 receptor. And therefore, the endocannabinoid really regulates tightly the immune system, not only by regulating their phenotype, what kind of cytokines, the release, they even regulate it's called phagocytosis, how they gobble up those bugs when they're fighting those infections. They regulate how the immune cells travel in the body. That's similar into neurons, developing neurons.
Right.
They're involved. So they have these really fun, and they regulate, I don't know, actually, Giovanni should check out if immune cells have CB1 receptors in their mitochondria, because that would be fascinating. Because we know mitochondria are so important for immune responses. You need the energy. So truly, endocannabinoids have this fundamental function. They regulate cellular function at multiple levels, somewhat similarly between cell types, proliferation, migration, selectivity of functions, neurotransmitter. They have a lot of CB1 receptors. But so yes, that opens up actually a new question, and there's an entire field there, which is there's many diseases in the periphery that can we actually develop therapeutics that are targeting the endocannabinoid signaling system and can help with those diseases, liver cirrhosis or things like that, autoimmune disease. That's how I started working on neuroinflammation when I started my lab, is because patients that have multiple sclerosis, back in the 90s, that was one of the diseases that was the most reported beneficial effect of cannabis use was the patients with multiple sclerosis. And therefore, that's how I started thinking, well, maybe it regulates the immune system. It dampens, because multiple sclerosis is an autoimmune disease. The disease is too strong. And therefore, let's try to dampen it. And that was the idea. Let's try to target the endocannabinoid system, CB1, CB2, to calm these immune cells in those dramatic autoimmune diseases.
And we've talked about how these endocannabinoids are deployed in a activity-dependent manner. They're made on demand. They're regulated in a way that, you can use it at this synapse over here when it's needed, but not the synapse over here. And so there's this really nice spatial and temporal regulation, this on-demand regulation of when these things get deployed. At the same time, we've talked about things like FAAH inhibitors, inhibiting these enzymes, which sort of determine the rate at which these signals are broken down. So even though there's this kind of point-to-point regulation and this very dynamic regulation of endocannabinoid signaling, there's also sort of just a background level or background tone that we can talk about. Endocannabinoid tone is a concept that sometimes you hear people talk about. What is that, and how much does the overall tone of endocannabinoids differ moment to moment in an individual or between, say, two individuals in a population?
So while we know much more about on and off production of endocannabinoids, because that's where neuroscience pay attention on how does it regulate neurotransmission, we know much less about the endocannabinoid tone. The way to think about it is that it's going to have a very different biological function. Endocannabinoids are produced in an activity-dependent manner, and they're going to regulate the cellular function over a small amount of time. I'm going to shut off this neuron for a while because it's too active. Whereas the endocannabinoid tone is going to tonically activate those receptors and therefore really change. So it's really, I think the only difference is basically the temporal activation of the cannabinoid receptor. Does our physiology want it on and off because we need to regulate neurotransmission now? Or are we going to regulate the tone and therefore change the overall phenotype of the neurons on a longer term? I'm going to activate these receptors maybe on the cell soma directly. So for example, there's a really fascinating, again, another function of endocannabinoids to help the brain, for example. So when you activate those CB1 receptors that we talked about on the main neurons, not the presynaptic, just on the cell body, they go and they change the phenotype, they change gene expression. One of the genes that they actually upregulate is brain-derived growth factors, BDNF. That's very valuable for the health of the brain. So here's actually one situation where you would imagine, although it's not been demonstrated, but if you put the building blocks together, that's the most likely hypothesis, is, for example, a higher tone of endocannabinoids will activate more CB1 receptors on neuron cell soma and therefore have more BDNF and foster neuronal connectivity and things like that. So that would be one biological function that would separate the tone of endocannabinoid versus the on-off, and I regulate neurotransmission. The tone probably regulates much more the phenotype of the neuron over a long term. And that's where the concept that you were referring to, changing our diet, right?
Right,
I would imagine that changing the diet will have an effect predominantly actually on the tone. And you might have, more endocannabinoids and maybe a healthier brain. This is all me putting pieces together. We've never proven this, but it would be a cool experiment to test.
Yeah. So like at any given moment, of course, like there's going to be highly dynamic changes that are highly context dependent in where endocannabinoids are being deployed and how many are being used. But that's all sort of riding on top of this general background tone that we're talking about. And you're saying that perhaps it's not proven, but perhaps changes in diet and things like that might slowly change that background level of tone over time. But on top of that, you're always going to have these sort of bigger, faster changes that are more dynamically regulated.
Correct. And the parallel is dopamine. Dopamine has also a phasic activation and then a tonic activation. And those two things actually do different things on the dopaminergic receptor. So I think this is a concept or a mechanism that biology uses quite often. Am I going to produce this neurotransmitter for a long time or just on and off? Probably both.
Yeah.
Because they have different functions.
Right. You can use the same signaling molecules, the same
absolutely
items, and you can change how they function or what they do by playing with either a slower time scale or a faster time scale changes in the signal.
Yeah.
What about, so like just based on your knowledge as a basic researcher and the general, what we know generally about endocannabinoid signaling and all of the complexities involved, what's your general take on the rise in THC consumption, exogenous cannabinoids that we've seen, for a number of years now? I forget some of the exact statistics and stuff, but we know that, cannabis use is up, at least in some populations. I believe some populations are now consuming cannabis more often or more overall than even alcohol. What are the pros and cons here, both in terms of the overall amount people are using and also sort of the differences between adults and younger individuals?
Absolutely. So that's a really good question, and there's a lot to say about this. I was actually involved in the task force to implement I-502, the legalization of cannabis in Washington State. And so I was able to actually be really in the field, understanding how new legislation is actually put together. The biggest aspect to think about there's several aspects. And we need to differentiate THC versus CBD. I'm not sure if we want to go after CBD, but if we focus on delta-9 tetrahydrocannabinol, THC, the first thing to consider is the age of consumption. And because we know there's actually really no doubt that there is a vulnerable population, a subset of adolescents that are very vulnerable to using cannabis during adolescence. Now, the question always becomes how much and how often, because everything at very high dose and all the time will actually be bad on your body, especially THC. So the fact there's either a legal system, you make it illegal for adolescents. That's one way to think about it, absolutely. The other way is to educate adolescents and let them understand that this is actually detrimental for their development. The good news is that there's actually an age where the detrimental effect is not as strong. I would say after 25 years old, when your brain is fully developed, then people start using cannabis and there's less impact on brain development because the brain is already developed. And maybe there might actually have some positive effects. It might reduce stress levels a little bit. It might, temper inflammation, sometimes help you sleep for some individuals. So there's really a period during development where we know it's going to be bad for the brain, and then a period where we can actually start thinking about good regimen. How much am I going to take? How often? And that might become medical. So that's one aspect, the regimen and how much. And now we can get into the products, the cannabis-based products. So what has happened? Oh, yes, that's right, because you also interviewed Ryan Vandry for the
Yeah.
PK.
Yeah.
He's excellent, and he's asking very important questions. So depending on the product, are you going to smoke it and therefore it's going to go into your brain on and off very quickly? Are you going to only smoke a joint, or are you actually going to dab 35% THC versus 95% THC? Plus, dabbing is going to go up and down very quickly into your brain. That is the one first criteria that will increase the likelihood of getting addicted to a drug. If your dopamine goes up fast, up nicotine, up and down very quickly. So dabbing THC, high concentration of THC very quickly will induce a very high increase in dopamine and their potential addiction, cannabis use disorder.
Yeah. And let's define and talk about that just a little bit for people, because it's important. And there are very different viewpoints and biases that people have out there. You know, when I worked in the marijuana industry, this was a very polarizing subject and very difficult to address, because some people would insist, you know, cannabis is not addictive. It's not addictive like other drugs. And some people would say, well, yes, it is. It is habit forming. Addiction, dependency, habit forming. Like, what do these terms mean to you? And from your perspective, you know, what is the habit forming potential of THC in particular relative to other drugs that we can talk about?
So the first thing is that both are right. It is true that some people get addicted, and it's true that some people don't get addicted. Thus, the concept of vulnerable population. Some people are more vulnerable to drug addiction than other ones. The precise reason why they're more vulnerable is something that the field is working on. Genotyping. Is there a genetic makeup that makes this poor individual more likely to become addicted or not? So I think both are right. I think some people get addicted and some people don't. What is the term of addiction in the field of cannabis? It's related to cannabis use disorder. So in the DSM-5 now, there's some criteria. It's basically a new disease, a new syndrome that has been discovered because of the rise in the consumption of products that are very potent in THC. The more potent, the more you consume, the more likely you're going to have the cannabis use disorder. Within the cannabis use disorder, there's multiple criteria that the patient needs to have to be considered having cannabis use disorder. One is not being able to stop. That's the definition of addiction. Even though I know that it's not good for me, I'm not able to, I'd rather continue smoking. That would be one criteria. How addictive is it compared to other drugs? It comes back to how much dopamine is produced in the brain and how quickly. So nicotine is super addictive because you have a very quick rise. Cocaine is super addictive because you have a big rise. THC is more and more addictive as you start using the dabs, for example, because now you have 95% of THC on and off. So that's how I think about it. It's like really the amount of THC, how often, and only of a subpopulation, a vulnerable population.
Yeah, and I think it's worth dwelling on and really emphasizing this idea of vulnerability in discrete subpopulations, because it really is true. You know, I've done a couple episodes with Christian Lucier, who does excellent work here. And just to give people a sense for this, like, you know, you can give rats cocaine and some of them get addicted, but some of them don't. And it's actually, It's not just a random subsample for reasons that are still somewhat mysterious. there are discrete subsamples of individuals, whether it's rats or humans, that are just more prone to addiction to virtually anything, it seems in some cases, than others. So it's not that, everyone becomes addicted if you consume enough, or no one becomes addicted because it's a non-addictive substance. It can be that if you consume enough, and on top of that, you're this sensitive individual rather than someone else, you can develop dependence or something like that. But
Absolutely.
there's
And the fact that we actually see that in the preclinical rodents in
yeah,
rats
yeah,
is actually a power for us to understand what is the difference, because we can actually do some gene sequencing and
yeah,
try to understand that.
yeah. What's kind of fascinating about that work is in rats and in people, the numbers kind of shake out the same. It's like something like
Correct.
roughly 1 in 5 rats will become addicted to cocaine if you just give them a bunch of cocaine over and over again and nothing else to do. So it's like 1 in 5. But why that 20% and not the other 80%? Nobody knows. But it turns out that the numbers are kind of similar in humans too. It's like this 1 in 5-ish percentage of the population, for that
I
substance
agree.
at least, that's sensitive. And is it a birth order thing? Is it the way you grow up? I don't think anyone really knows yet, but
I would argue it's probably really fundamental biology, because
yeah.
rats in a lab are so similar.
Right, right,
And then we have exactly the same thing in this really diverse population of
right,
humans. So it's
right.
probably something very fundamental.
Yeah, Christian Lucier had the same intuition, because that is a huge difference. Like, these rats are almost genetically identical, but humans obviously aren't. So I don't know what the answer is. I think he's working on some hypotheses that have to do with like birth order and how much
Oh,
of the nutrition
nice.
you get early in life as like the last pup born or whatever. But TBD on that.
Yeah, I'm excited. He's an excellent scientist. I can't wait to see what
Yeah.
happens.
What are some of the things that you're working on today, or that just the field in general is working on, maybe using some of these new sensors, that you think are going to produce some exciting new insights relatively soon in the future?
Well, yes. So one of the biological questions that we're testing in my lab is really to try to better understand the effect of THC on mouse behavior. So we have a couple of papers coming out now that have been accepted. One, I'm excited, coming out in Nature, that actually will describe multiple things. Uh, the first thing is that we actually studied a very specific behavior in the mouse, which is the mouse approaching a sucrose um, um, bottle, so that it's a reward. So the mouse is very excited. It comes close to the sucrose bottle and sips a little bit of sucrose, and that's a reward. And you can actually train the mouse in the Pavlovian reward training, so they get actually excited by the reward coming up, because there's a little light that opens up. So what actually drives this behavior, which is really fundamental, especially if you think about reward and addiction, how can we understand this behavior? So we actually looked at the endocannabinoid signaling system in specific brain areas, in the thalamus that projects to actually the nucleus accumbens in the brain. The nucleus accumbens is really an area of the brain that's involved in drug addiction. And what we discovered is that these neurotransmitters that go from the thalamus and project to the nucleus accumbens, the neurotransmitter release of those is regulated by endocannabinoids, also CB1 receptors. So we actually discovered a new function for the endocannabinoid signaling system in reward, which is to simply control the approach and going after. So that's the first part that we discovered. How did we discover that? We developed a machine learning pose estimation algorithm. So in this time of AI computing, I love new technology. So we got really interested in monitoring those little mice in a linear track and tracking them, just like when you do facial recognition, where you know where the eyes are. We have little dots on the mice, on the tail, on the nose, and we look at them as they travel in this linear track. They walk, they stop, they groom, they rear, they explore. It's fascinating. And then there's the sipper, and they go and they sip the sucrose. So we can actually, with a video camera, monitor these mice And then with this AI machine learning approach, basically analyze what they're doing. And we can say exactly how quickly it approaches the bottle of sucrose, how quickly it disengages, and all of these behaviors.
I see. So
And
using these types of tools, you can start to ask questions like, okay, if we put a bunch of mice in the chamber, they might all approach the sugar water sometimes, but some do it more often. Some do it more
correct.
quickly. Some are more interested in doing other things. And you can sort of ask, how do things like endocannabinoid levels relate to those individual differences?
Correct. So the way we know that is now we can actually add those sensors that I talked about in these mouse brain, specifically, for example, in the thalamus, that brain that I talked about, or the nucleus accumbens, and look at how these endocannabinoids are produced in an activity-dependent manner. And it turns out that as the mouse approaches the sucrose bottle, there's these endocannabinoids that are produced for a couple seconds. And the idea, and that was another part that we kind of did in the paper by electrophysiology, is that these endocannabinoids in this brain area are going to activate those CB1 receptors with specific dynamics and regulate neurotransmitter release, reduce a little bit of the GABA transmission, reduce a little bit of the glutamate, and that will participate in the neuronal network that will control this behavior of approach. So that was the punchline of this paper, is really discovering the fundamental function of, in this case, it's 2-AG, the second endocannabinoids that's mainly produced. One new function of 2-AG in the brain, which is to actually control reward approach. And that obviously brings up a new question. Can we use this new understanding to develop therapeutics for drug addiction or for cannabis use disorder, now that we know that these endocannabinoids are produced there at that moment?
Yeah. And so it's just worth emphasizing the tools you're talking about. So you not only have the machine learning based video tracking tools that allow you to quantify and record what the animal's doing at the overall behavioral level, but some of these new sensors are allowing you to very specifically record endocannabinoid dynamics. How quickly are endocannabinoid levels going up and down in particular parts in the brain? So you're not just looking at some sort of very, very fuzzy high level signal. You're actually getting quite specific with this.
And very temporally specific, in a very time dependent. And at the same time, we're also measuring neuronal activity. So like that, we can actually connect what we talked about. There's more neuronal activity, there's probably more calcium, and therefore more endocannabinoids that are produced that are going to regulate those CB1 receptors on nearby neurons.
What about, so cannabinoids have powerful effects. Exogenous cannabinoids have powerful effects. Endogenous cannabinoids have powerful effects. They're tapping into this sort of homeostatic regulation that's really fundamental. And this is why, cannabinoids have so many different effects. One of the major side effects of THC, as we mentioned earlier, is the munchies, right? Animals tend to get hungry, they eat more. And yet, there's this interesting and seemingly paradoxical situation that you mentioned earlier where, yes, they stimulate feeding behavior and the munchies, but long-term THC users tend to have lower BMIs, lower body weight. So what's going on there? Well, that might connect to something that I also think is interesting, which is that there used to be, for a little while in Europe at least, this CB1 receptor-based therapy for obesity, basically. It was a drug called Rimonabant, which antagonized the CB1 receptor and caused people to eat less. This was before GLP-1s were developed. You've probably never heard of Rimonabant, most of you, because it got pulled from the market. What was the Rimonabant story, and what does that sort of tell us about how powerful things like the CB1 receptor actually are and how difficult it is to play with them?
Yeah, the hypothesis there was very simple back then. Sanofi Pharmaceuticals in France. The idea was, well, if THC is an agonist at CB1 receptors and produces munchies, we're going to do an antagonist and it's going to block the munchies. And therefore, that was Rimonabant, or SR1, that people talk about in the field. And that molecule actually went all the way to being accepted in Europe. So it passed all the clinical trials, the safety.
Yeah.
And
but
then
it actually made people eat less.
It actually made people eat less. You would be on this drug and you would show the patient a cookie and they wouldn't eat it. Like, when does that happen? Only when your CB1 receptors are blocked. So then it's accepted. And then what happens, when you do a phase 3, you have thousands of people. And then when it's accepted, now you have hundreds of thousands of people. And therefore, the likelihood of identifying a side effect is increased because more people are using it. And what happened, and actually it didn't happen right away, it happened after a couple of years, if I remember well. Some patients were getting depressed because actually CB1 receptor controls mood. And therefore, if you break the CB1 receptor, you don't have the reward aspect of going after your cookie. You might actually become more depressed. That would be to the point. Some patients were so depressed that they committed suicide. And that's when, I think it was like after the third or fourth suicide, then they took the drug out of the market because the side effect is too dramatic if people make. And that shut down so many drug development projects trying to target the CB1 receptor in the brain for society to stop eating. But then what happened is the field continued to work. And then what we discovered is the CB1 receptors on the liver. And the CB1 receptors on the liver are the ones that regulate metabolism, obesity. And therefore, now there's a very simple trick that you can do in drug discovery is make a CB1 antagonist that does not go to the brain. That's very easy. You put a molecule and then it won't pass the blood-brain barrier. So now you're avoiding the depression, the suicide, because you're not targeting the CB1 receptors in the brain, but you're still targeting the CB1 receptors on your liver that actually controls metabolism. So that's where the field is at.
Have people actually done that?
Yeah, that's happening right now. There's some molecules that are getting closer to human clinical trials, and those CB1 receptors do not pass the blood-brain barrier.
So we've tested those in animals, CB1 antagonists that do not cross the blood-brain barrier.
Correct.
And what is the basic overall effect in those animals?
Well, they eat less. They
Got
eat
it.
less.
Absolutely. So you get a similar Rimonabant-like effect in terms of eating less, but they don't show any indications of anxiety or depression-like behavior.
Correct.
Okay.
And actually, this is a field that I follow very on the sides only, because then the question is, I understand how if you activate CB1 receptors on the liver, you're going to change metabolism. Okay, that's fine. How I'm going to digest food, but what about the motivation to get that cookie? Is it really only the CB1 receptors in the brain? Well, no. There's actually some research that shows that when you get hungry, there's actually another one of those lipid signaling molecules that is produced by the liver or the digestive system, goes into the blood. It's a lipid. It passes the blood-brain barrier, goes to your brain, activates its own receptors and say, go eat. So now the CB1 receptor antagonists that are peripherally restricted, they regulate the metabolism in the liver, but at the same time, those lipids can still go to the brain and tell you, oh, go ahead and eat.
What are those lipids?
Oh, I forgot what they are, but they're also signaling lipids. Yeah.
Got it. Yeah, signaling lipids, not unlike cannabinoids, but they're different ones. They're made in the liver. They go through the bloodstream and they actually pass the blood-brain barrier, and they can affect motivation to eat, even though the signal arises in the liver.
Fascinating. Absolutely.
Wow. So as a cannabinoid biologist, knowing the history here with Rimonabant and how cannabinoids work, we've got the GLP-1s now. Lots of people are using them at this point. We're coming out with new versions basically every month, it seems. So this is going to be a huge area of growth, both in terms of the number of people using them, but also the diversity of the specific versions coming out. Are there any things that we should look out for as we go into the second, third, fourth, fifth year of people using GLP-1s in terms of them potentially having effects on mood and on motivation akin to what the cannabinoid-based drugs did in the past? Or are they likely working in such a different way that those types of side effects are unlikely to be seen?
I think they're working in a very different mechanism, and therefore the results are going to be very different. I think this GLP-1 field is fantastic. It's so interesting, and it has a lot of potential indication. We started with obesity and food intake, but actually it turns out that that might be also potential beneficial for some addictive drugs like, nicotine and ethanol. So it has that parallel with the cannabinoid signaling system that it regulates, food intake and reward. But I think they're going to be, because the receptors are very different, because the receptors are expressed in different parts of the brain compared to the CB1 receptor. I think the GLP-1 drugs are going to be having different effects. And plus, it's only the beginning of this entire field. Now they're optimizing, like every field in medicine, transformative. It always starts, and then we have a little bit of toxicity, and then we optimize, and we can actually develop a completely different therapeutic approach. One thing that this is anecdotal, but I thought it was fascinating. So anecdotal, N equal 1. This individual in California was taking GLP-1 drugs to lose weight. And then it's the birthday of their kid, and there's a beautiful cake, and they want to eat the cake of their birthday, and they don't have the motivation at all. And that individual actually used cannabis, got the munchies, and was able to actually enjoy the cake. So that to me is fascinating, because it really tells me that those signaling systems are actually very different. And might be, it's just an interesting tidbit. I'm not sure.
Yeah, because the person wants to eat the cake. They want to lose weight, obviously, but they want to participate in the ritual of the birthday.
That one day is okay.
Yeah,
exactly.
Yeah. So you might have a temporary tool, in this case, cannabis.
Exactly. An exit.
So what do you want people to know? What do you want people to take away about cannabinoids, generally speaking? And I'll let you decide what you want to say. Endogenous cannabinoids, THC and exogenous cannabinoids. But what's something that you want to really emphasize or reiterate from people related to the biology that we discussed today, that are listening and that are interested in cannabinoid-based therapies or substances of any kind?
Absolutely. I think the field has come a very long way. And that a couple things. The first thing is, when I started studying the cannabinoids 25 years ago, signaling system, I spent a lot of my time explaining to people that it has some medical properties and not only the psychotropic effect. Now the field has recognized and understood that cannabis has medical properties. And I spend quite a bit of time telling people that it's not going to cure everything. It's going to be very specific. So that's one thing that I think I'd like people to understand, is that cannabis, we used to talk about it as the drug for the potheads. You want to smoke out and watch a movie and things like that. That's fine, or mind altering. That's one way to think about it. But I think the future, we will maybe even forget that aspect and talk about cannabis as a transformative medicine, because it was that the research that we are doing right now resulted in medicines that are beneficial or might even cure some diseases where we had nothing. The first example that comes to mind is the epilepsy field with the Dravet syndrome. When cannabidiol was accepted by the FDA, that was a landmark, because these poor little kids, they have really pretty much nothing to help with those seizures. And then you have this CBD that transform a little young girl that has hundreds of seizures per week to only a couple seizures per week. That's a life-changing experience. So I think we're at the very beginning. And I've been in the field for so many years that I've seen it evolved. And the beginning was, okay, let's figure out the receptors, okay? And then let's figure out the enzymes, okay? And now where we're at is small molecules in clinical trials that are very promising for specific diseases, which is why I think the cannabinoid field is going to generate some transformative new medicines that are going to help people.
All right, Professor Nephi Stella, thank you very much for your time.
That was great. Thank you.
Hey great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.